right, this will be all about DNA technology. So when we talk about DNA technology, it's also referred to as biotechnology. It can be genetic engineering. There's a lot of different things that get kind of thrown into it and talked about kind of the same way. So biotechnology, DNA technology, it all refers to technology that manipulates DNA, and really it's the purposeful manipulation of DNA. So often these procedures can be referred to as genetic engineering. DNA is genetic material of all living organisms, and we know, we've learned that how it's replicated, we've learned how protein synthesis takes place, and we know that every living organism has DNA in it. And we also know that all the organisms use the same genetic code. Well, it's because of this that we can use DNA of one organism to manipulate DNA of another organism. So therefore, genes from one organism can be transcribed and translated and put into another type of organism. And we do this. A lot of people are familiar with some of the processes, but this is done every day. And it's been done for some time. We manipulate genes. We've just done it quite a bit. Human and other genes are routinely put into bacteria in order to synthesize products for medical treatment and commercial use. Human insulin that we just learned about in the DNA and RNA part can be made synthetically now. Human growth hormones, vaccines, and others are produced by bacteria. And I guess I misspoke a little bit, because this is not a synthetic process. This is a natural process. We're just manipulating it. So human insulin can be made through manipulation. There are different types of DNA. Recombinant DNA refers to the DNA from two different sources. So individuals that receive genes from another species are referred to as transgenic. So often you've heard of transgenic crops and different things like that. So there are viruses that are out there that can contain genetic material that are not living. However, they can be host cells, or host cells are required for their reproduction. Viruses have a nucleic acid or genetic material. As you remember, we talked about way back in the beginning of the semester. So viruses can infect animals and different things like that in other organisms. Some viruses are DNA, some are RNA. This is what the structure of a virus looks like that they have there. They have the envelope. They can have spikes, they don't have to have. They all have a capsid, and they have either DNA or RNA inside. The capsid can be in different shapes. It can be helical, polyhedral, or complex. With viral reproduction, what we have is the genetic material enters the cell, it's replicated, it's transcribed into mRNA, and it's translated into the proteins by the host cell. This process, the host cell uses the genetic instructions of the virus to make more viruses. So it's basically the virus telling the other cells, hey, make more of me because you should like me, even though I'm bad for you, but I'm in control now. So if the viral genetic material is RNA, then a DNA copy must first be made for the transcription and translation to occur. The DNA copy of the viral RNA is called cDNA. The viral RNA, or the cDNA, and then the mRNA, and a protein. So it goes, you have the viral RNA, it's coded into a DNA molecule, then it is translated into an RNA molecule, then it can be transcribed to a protein. So we also have these bacteriophages. Now, a bacteriophage is a little bit different. A bacteriophage is a virus that has infected a bacteria. So we can use these to get different targeted things into an animal or anything like that. So it's not surrounded by a membrane, and it has an animal-infecting virus. The virus is attached to the bacterial cell. A viral enzyme digests away part of the wall, and then the viral DNA will enter the host cell. Once it's inside the host cell, the DNA is transcribed, translated, and replicated. This translation produces protein coats on the enzymes to construct new viral particles. Then the viral DNA is replicated. The protein coats are then disassembled, new viral particles are released, the host wall ruptures, the newly formed viruses go out to infect other cells next to it. So this is kind of describing it here as we have the first step here. It attaches on, sends the DNA in. The DNA makes other little particles in here. The other particles then get synthesized and assembled and everything together through that process. They burst the cell wall and the viral particles can go out to do it again. So upon entering the cell, the viral DNA may instead become integrated into the DNA. It's replicated along with the host DNA procedures. Eventually it will become transcribed and translated. This is like, think of this, the herpes viruses are DNA viruses. They implant themselves into the DNA of the host. Then we can have retroviruses. Now, retroviruses contain RNA and an enzyme called reverse transcriptase. This makes them a little different. So we kind of have to work. They have this enzyme that makes them a little more specific and able to do things. And the new DNA that's produced from the RNA template is called the cDNA. We have that as well. The cDNA becomes incorporated into the host DNA. Then it's a prophage. The new viruses escape by budding. But what happens, this reverse transcriptase keeps going back. So if our body was to synthesize out the DNA, it keeps going back and implanting its RNA molecule, making the new cDNA each time that it does so. We also have vectors that we have here. Vectors are used to transfer genes into a host cell. So this is using something to transfer into a host cell. That could be a plasmid. A plasmid is a bacteria that is used to transfer. A plasmid is an extra chromosomal structure that we can put into bacteria. Plasmids and viruses are usually these vectors. A vector must be capable of self-replicating inside the cell. Well, viruses can do that. Plasmids can also do that. Viruses are the vectors of choice for animal cells. So, because marker genes can determine if the vector has been taken up. Viruses can accept larger amounts of DNA than plasmids. Plasmids is one single circular chromosomal structure. Viruses can have vast amounts of DNA. And once the virus enters the host cell, it produces foreign genes. It's considered to have been copied or cloned. Now, once we've done all this, we have to figure out how we can put the DNA or the genetic material into a new organism. We do this with restriction enzymes because we can't just take a pair of scissors and cut DNA. We know that DNA works off of frequencies, and the DNA frequencies we can create breaks in the DNA frequencies or in that code. So what we can do is we can use that code and then we can use that frequency and those codes to then be able to cut the genes wherever we want to and put them back in, because the restriction, that's where it gets restricted at that frequency, and we can use it to cut there. There are hundreds of different restriction enzymes that have been isolated. However, we're going to talk about a few of them here. Each restriction enzyme cuts the DNA at a specific base sequence. Remembering that those base sequences are complementary to one another, it's going to cut at the same position on both strands between those specific frequencies. So the EcoRI always cuts DNA at the GAA-TTC frequency. So you can see this is the 5'3", this is the 3'5". It cuts right there. You can see it cuts at the GAA-TC frequency. So it cuts right there at the same time. GAA-TTC, and it cuts it. The sequence of GAA-TTC appears 3 times in the DNA. As a result, it's going to be cut into 4 pieces. So you have the GAA-TTC, you have and you have it each time, the GAA-TTC. So it's every time that it shows up, it's going to be cut. The resulting fragments may be left over here that aren't those. So there are other restriction enzymes out there that cut at different sites. Some of them are below the BAM-HI, Hindi, the PST, HAE, all have different cutting sites. So you can see that once the fragments have been cut, they have what's left over and is referred to as a sticky end. The sticky end basically just means it's an end that's open to attaching on to other fragments of DNA at that point. Sticky ends have complementary base pairs so that they can rejoin. Those sticky ends are here. See these open fragments here? So they're open to rejoining at those same complementary pairs. So if the vector or the gene to be cloned are both cut in the same restriction enzyme, they will have complementary sticky ends. After cutting, the two DNA samples can be mixed. So you have to use the same restriction enzyme on both organisms, then you can stick them together, so to speak. Fragments with the complementary sticky ends joined together to form what's called recombinant DNA. This contains a gene from one vector and then the gene to be cloned. The enzyme DNA ligase seals the fragments together. Bacteria such as E. coli are capable of taking up DNA from the environment, so this is where we can use the process of transformation. It's actually a little bit easier that we can use this transformation, and we have to use calcium chloride and heat shock and things like that to make the E. coli more permeable. It causes it to kind of open up, and then we can modify the plasmids and we can get them to take in. So we also have many different genomic libraries that we have out there. This is all the genes of a particular organism. Bacteria or virus vectors can be used to store fragments of DNA from another species. The DNA is cut into fragments, and different fragments can be inserted into bacteria or viruses. Then they can be stored in a library, or they can be used in different organisms. We have the process called PCR. PCR stands for polymerase chain reaction. This is used to make copies of small DNA pieces, and it requires primers, the DNA polymerase, and nucleotides, because remember, DNA polymerase was part of that genetic replication. So there are primers of about 20 nucleotides in each of these short chains that are complementary to the region of the DNA, and it amplifies that region of DNA, essentially. The DNA polymerase cannot continue to process unless it's already been started by the primers. Nucleotides are needed because DNA is composed of these nucleotide building blocks. So essentially what we have is we have our DNA that's in the small pieces. Then we have our primers that begin. We have our enzyme DNA polymerase, and we have our nucleotides free out here, and we can put all of these back together. So you heat the DNA to 25 degrees Celsius to separate the DNA strands. Okay? Then you attach, after the strands have been separated, the DNA is cooled down to about 50 degrees so the primers can attach. The temperature is then raised back to 70 degrees so that the polymerase, the enzyme, will attach and copy the strand. So that's essentially what's going to happen then. It's going to bring those primers in. The DNA replication process repeats itself. The solution is then heated and cooled at regular intervals until the process is complete. So we're forcing the DNA to make those complementary strands of one another. We then have the DNA fingerprinting, or RFLP analysis. In RFLP, the DNA of an organism is cut up into fragments using restriction enzymes that we already learned about, and a large number of short fragments of DNA in the process we just talked about. No two individuals have identical DNA. No two individuals will have the same length of fragments. So this is whenever we want to run a DNA fingerprint. This is through the process. You've probably all seen it in crime shows and different things like that, where they have different frequencies. So everybody, if you have a different length in each of your DNA fragments, they're going to stop at different locations, and in stopping in different locations, that creates a genetic fingerprint. We can figure out your genetic fingerprint through this process called gel electrophoresis. This is where we take those separated fragments and we kind of isolate them out according to size We run them through electrical currents in a sheet of gelatin, and the negative DNA is negatively charged and it moves through from pole to pole. Well, once your gene lines or your length of your fragments reaches their frequency, they stop. As they stop or get restricted, everyone's will stop at different locations and different genes do different things. So this is kind of what it looks like. You put it in the gelatin and you move it through the positive power supply, put the power supply, and it runs through there. The smallest fragments will move the fastest, and they're able to move through the pores of the gelatin faster. Bands will start getting produced where the fragments start to accumulate. The shortest fragments will accumulate near one end of the gelatin furthest from the well, and the longer, slower moving ones will remain at the other end. The DNA bands must then be stained through some different processes. We've always used ethidium bromide, which is a toxic substance, but it still works, and it makes them glow under a black light, essentially. So you can see that the faster moving bands are going to end up here. The slower moving ones are going to end up down here. So other practical uses for genetic engineering: E. coli is used to produce proteins such as insulin. We can make the insulin that we use and insert into people today. To recover the product, E. coli has to be lysed, so you kill the E. coli, or the gene must be linked to produce a natural secretion protein. So you're not actually using the E. coli, you're just using the DNA from it. Yeasts can also be genetically engineered and are likely to secrete a gene product continuously. Mammalian cells can be engineered to produce proteins such as hormones for medical use. We can use cells to make hormones to put into people, growth hormones and things like that. Also, this is where we can make certain steroids and things like that for breathing. Plant cells can use a plasmid to make agrobacterium. Plant cells can also be engineered to produce plants with new properties, such as Roundup-ready soybeans, different things like that. Pseudomonas bacteria are used to produce Bacillus thuringiensis, or BT corn, that you've probably heard of if you've heard of that. Takes the toxin against insects, producing a natural insecticide, BT cotton, BT corn. It's out there, and it kills insects whenever they get a hold of this material, this toxic agent. Other uses: animal viruses have been engineered to carry a gene from the pathogen's surface protein. The Human Genome Project was a project where we completely mapped every entire gene in the human body, all the chromosomes, all the genes. We knew everything about it. It took several years to do. This has helped us to provide tools to diagnose and repair genetic diseases. Recombinant DNA can be used for genetic fingerprinting so that we can solve crimes, we can tie people to different genetic traits, and we can do genetic testing. Gene therapy also could be used to cure genetic diseases by replacing defective genes. Bovine growth hormone doesn't get used much anymore, but it increased milk production in cows by about 10%. There are lots of questions out there with safety and ethics. We probably all remember Dolly the sheep and how she was cloned, and nobody liked it because then they were going to be cloning humans, and it was going to be awful, and so on and so forth. So organisms that are intended to be released into the environment may be endangered. So, we could take organisms that have been endangered and we could clone them. We're not talking about Jurassic Park, for the record here. There is little legislation in the use of genetic screening right now. I think this is probably the biggest thing: is we don't have a handle on it. We don't know what all the possibilities are. People could, you can already clone your pets if you really wanted to, things like that, I guess. So, but there are a lot of ethical questions of whether we should be modifying the genes of humans or not. It raises, designer babies, and all kinds of questions get raised. Genetic screening and gene therapy can be expensive. It's not available to everyone. Also, biological weapons could be used creating biotechnologies. So that is the biotechnology discussion for today. Thank you.